Bismillah Rahman Rahim. Welcome again, guys. Welcome to another video lecture by Biology. Today in this video lecture, we are going to talk about screening of cDNA library by oligonucleotide probes. Okay. In the previous video lecture, if you guys remember, we talked about the screening of genetic library by colony hybridization. Actually, we are going to talk about the screening techniques in step-by-step video lecture. Now, in the previous video lecture, we talked about that technique, and before that video lecture, we talked about the screening technique and different ways of the screening. Now in this video lecture, we are going to talk about screening of the cDNA library by oligonucleotide probes. Now, how we can perform this technique to find out our vector having our target DNA or gene of interest. We can perform this screening that we should make a probe DNA or RNA sequences. That probe DNA or RNA sequences should be possible by knowing the protein or the peptide sequences which are produced from your desired genes or from your desired sequences. Okay, so due to this way, you can design your oligonucleotide probe. Okay, so how we can do that, what are the stages, what are the steps are involved. But before that, you need to understand about the colony hybridization, because the stages or the steps which are involved in colony hybridization, each stages and steps, some of each steps are also involved in other screening techniques. So in other screening technique, the same steps, some of them steps are also followed. So you need to understand the colony hybridization technique. Okay, so it is present into my channel. I already explained it. You can go back to my channel, you can watch it. So let me clean the whiteboard and let me start next topic. Okay, so let me write screening of Now, how we can perform this technique? In this technique, we should know the peptide sequences. Okay, let's suppose in this case the protein are produced insulin protein. Okay, let's suppose. So insulin protein are produced in this case. So we better know about that, what kind of proteins, what kind of amino acids that are usually found in the protein. We should know about each of the amino acid due to which the protein are made, means insulin are made. So we should know about these sequences. Once we know about each amino acid sequences, then the DNA, possible DNA sequences which will made according to this will be very easy. And after this DNA, we are making a complementary DNA of this possible DNA sequences. And this complementary DNA, we are using it as a probe, which we call that oligonucleotide probe. Why? Because we can make it by this peptide sequences. Okay. Now it is using as a probe, and we apply it on the colony, and then if any one of the colony catch it, and any one of the colony can show some signal in the form of color, in the form of fluorescence, then it will show that this colony have all those bacterial cell, they contain all those bacterial cell which can carry your target DNA having the gene of interest. Now how we perform that, how exactly we are doing this process. So let's explain. So let's say these are the protein sequences. So all of you better know protein have two ends. In one end they have carboxylic acid, and in the other end they have amino group. So like that, COOH and NH2. Okay. Now after that, we should treat this protein with protease enzyme to break down or to degrade this. So we should degrade it by protease enzyme. Now this protein will be converted into small peptides, as you can see. So let's suppose we take any one of them, only we take one of the peptide. So let's say we take this one peptide for our to make the possible DNA sequences. So then this peptide, they have also, the carboxylic acid at one end and amino group at the other end. These peptide sequences are then, these peptide are then sequences according to Admon degradation process. So according to Admon degradation process, we should, make the sequences of this peptide. So for example, let's say these are the protein. So this is the peptide and it is no sequences according to Admon degradation. Now all this peptide is now sequences. This is amino. These are the amino acid, as you can see: phenylene, leucine, cysteine, lysine, tyrosine, methionine, histidine, gens, aspartate, and serine. All these are the name of the amino acid. So we make sure, or we know about the nucleotide, or we know about the DNA sequences, possible DNA sequences according to each of this. We know. Okay. So in this case, we should make the DNA sequences, possible DNA sequences according to this peptide sequences. So we better know that what kind of DNA sequences are found according to this peptide sequences. So let's suppose in this case we know about T, G, C, A, double T, A, C, A, T, G, C, A. So now these are the DNA, possible DNA sequences, as you can see. DNA sequence, and this is the these are actually the peptide sequence. Peptide sequence. Okay, now these are the DNA sequences. Once we make the DNA sequences according to noun peptide sequences, then we will make the complementary of this DNA sequences. So let's suppose the complementary of this DNA sequences are so we should make the complementary by using DNA polymerase. If this is the DNA sequences, so we use DNA polymerase to make a complementary sequences of this one DNA. Okay, so we should make complementary sequences like okay. Now this is the complementary sequences. Complementary sequence. Okay, and we should also add a radioactive substance with this sequences. Let's suppose iodine. We should add iodine with these sequences, as you can see. So let's suppose we label this complementary DNA with iodine. Now it become the probe, and this probe is known as oligonucleotide probe. Nucleotide probe. So we use it as a probe, and due to this way we can identify our vector having the gene of interest. These are the sequences, DNA sequences, which are actually found in our, these are actually the vector DNA sequences. Okay, so that's why we can make the complementary. How we make this DNA possible sequences? We can make this possible DNA sequences because we better know about the protein sequences. That's why we can make the possible DNA. Now with this possible DNA sequences, we make the probe, which we call that oligonucleotide probe, which is complementary to this possible DNA sequences. Now we use this probe or oligonucleotide probe onto the plate having all the bacterial cell. But before that, what actually we have, we should proceed all the steps which we performed in the colony hybridization. Again, let me explain here that what actually we have. Let's suppose this is the master plate. So I'm not going to explain it again, but let me tell you the basics here. So this is the master plate having all the colonies, as you can see. And here we will put, or we will press the nylon filter paper on the surface of this master plate. So let's suppose this is what nylon filter paper, which is pressed on the surface of this master plate. Nylon filter paper, and this is the master plate. After that, we get a replica, as you can see. The exact colonies on that nylon filter paper. After that, we should treat this nylon filter paper, or we should put this nylon filter paper into SDS solution, sodium dodecyl sulfate. It is a kind of detergent. Due to this way, all the colonies which contain the bacterial cell will be ruptured, and we have just the vector or the genomes of each bacterial cell. So we get this diagram, or we get the structure of each colony in this form. Let me draw. So let's suppose this is the structure of each colony. Now this structure can show that the bacterial cells are ruptured, and now these colonies have just the vectors and the genomes of the bacteria. Now after that, what we are going to do, we should fix it. How we can fix it? All of you better know. We fix all the colony by putting this nylon filter paper on a solid surface having temperature 80 centigrade, or we should expose this nylon filter paper on UV light, due to which the colony are fixed. Now after that, we take out this filter paper on the solid surface having the temperature is 80 centigrade or from the UV light. Now after that, this filter paper is, on this filter paper we should add the probes. So after that, we should add the probes. So probes should be added. Oligonucleotide probes. Now once oligonucleotide probes are added, so any one of them colony will be, show some detection. Any one of colony will become dark, or it become, it shows some changes. Some spouts will be detected on the X-ray film. So after that, what actually we are going to do, we should see this nylon filter paper onto the X-ray film. So this is, let's suppose, the X-ray film. X-ray film. So on X-ray film, what actually we are detected? We detected that one of the colony are become dark, or one of the colony are become, spouted, or it shows some signal, as you can see. And this signal is actually showing that such colony have all those bacteria. It is not a bacteria this time. All those vector which have your target DNA. Okay. Now then we will compare this X-ray film, or we will compare this nylon filter paper with the master plate. So let's suppose I am now comparing this X-ray filter paper with this master plate, and I will see that which one colony have such spouted, or which one colony have like this one characteristics. So this colony don't have the same characteristics. This don't have, this also don't have the same characteristic, but the middle one have the same characteristic, as you can see. And then I will make sure that actually this one colony have all those vector, and then I will separated this colony, and I will make another plate, and then we will use it for our purposes. For example, this is, let's suppose, the insulin producing vector, so we are using it for our diabetic patient. Okay, so then it is used for different purposes. It is not only here I just give one example of the insulin. So growth hormone is also, you can also take an example of growth hormone. So this is all about the screening of cDNA library by oligonucleotide probes. Okay, I hope you get idea about this video lecture, but if still you have any kind of question, you can write it into a comment. Inshallah, I will give you answer as soon as possible. If you like this video, make sure to hit the like button, share this video, and subscribe the channel to get more interesting videos like that. And stay tuned, because in the next video lecture we are going to talk about the immunological screening, which is very important. Thank you so much for watching.